My laboratory has always been keen to study problems that are clinically relevant. And in 2002, we studied the problem, how do we identify the women that need chemotherapy after the diagnosis of breast cancer? And the test that we developed, that is now known as MammaPrint, is a molecular test that identifies only those women that are at high risk that the tumor may come back. And as a consequence of that high risk, they should be treated with chemotherapy post-surgery.
And René recognized the opportunities of using genomics in the clinic, and then he rapidly took this platform in, combined it with the patient material we had here in the institute, and really developed this, what we now know as MammaPrint signature, in a very fast way. So I think there it illustrates his opportunism in recognizing this new technology, being genomics, to be valuable finally into the clinic, and then he takes the whole route very fast to actually make this possible.
In 2018, it's generally accepted that cancers are very different, even if they belong to the same cancer type. We call that personalized medicine. But in 2002, that concept was very novel. And as a result, we were among the first to show that through molecular analysis, you can see differences between tumors that appear similar under the microscope, but at the molecular level are truly different and therefore need to be treated differently. This is the MammaPrint test that we make available worldwide now through the company Agendia. It looks very low-tech because it's only a glass slide, but the reality is that there is more than 50,000 small pieces of DNA printed by an inkjet printer on this glass slide that allows us to measure the activity of all 25,000 genes in every tumor. And that gives us an overview of the molecular machinery that drives the cancer forward, and that allows us to draw conclusions as to how aggressive the tumor is going to be and whether a patient actually needs treatment after surgery.
When he sees an opportunity, he seizes the opportunity. And of course this is very important. I think René has a talent to really recognize those things that are really important and are most likely to bring success in the follow-up and potentially in the clinic.
We performed a clinical study that ran over 10 years in 100 clinical centers in 7 European countries, heavily financed by the European Union, in which we used the MammaPrint test on 7,000 breast cancer patients and followed them for a period of 5 years to ask whether the test is really accurate in predicting which women need chemotherapy and which ones don't. And that number that I mentioned, that 46% of women can safely forego chemotherapy, is the result of that study that we completed in 2016. A discovery is not worth much if it is not brought to patients. And we realized that to bring such a innovation to patients, you need to start a company. And Laura van Veer, my colleague, and I then started a company named Agendia for Amsterdam Genome Diagnostics, because we diagnose based on the genome cancer. And that company is now 15 years old. And currently we have 160 employees in Amsterdam and in the US. And we perform about 25,000 of these MammaPrint tests every year. And of course, our hope and expectation is that this number will grow quickly so that all women that are diagnosed with breast cancer will get this test to make sure that they are not over-treated with chemotherapy that they don't need. My long-term hope is, of course, that this generation can make cancer a chronic disease rather than a deadly disease. And I think we can accomplish that in the next 15 to 20 years. In the immediate future, I want to shorten the time that it takes to bring discoveries from the lab to the clinic. It's currently about 1.5 to 2 years between that I discover something in my laboratory and the first patients benefit from that, and I want to bring that period down to maybe maximum 6 months, if not shorter.